In this video, I'm going to continue the discussion of the HPT axis, that is, the hypothalamus, anterior pituitary gland, and ultimately thyroid axis. In the previous video, I talked about how neurons within the paraventricular nucleus release TRH, thyrotropin releasing hormone, into the first capillary bed of the portal system, which is located within the median eminence. TRH is therefore delivered down to the second capillary bed in the anterior pituitary gland, specifically within the pars distalis of the anterior pituitary, which is the adenohypophysis. We are zooming in here to look at a particular type of cell called thyrotropes or thyrotrophs, which are adjacent to this capillary bed. Right, so here we see one of these thyrotropes. We see that the capillary blood is flowing very closely to it, and so TRH is delivered and combined to the G protein coupled receptor on the surface of the thyrotropes. Okay. TRH activates a second messenger system, the DAG-IP3 system. As we know, IP3 triggers the release of calcium into the cytoplasm, and together with DAG, this activates protein kinase C. In this case, protein kinase C is going to lead to the activation of a transcription factor called PIT1. PIT1 is going to increase the transcription and ultimately translation of two different genes. The first one is coding for a protein that is 89 amino acids long. Here we can see a ribosome, which is translating the mRNA, which has been transcribed from the DNA over here. Also, PIT1 leads to the transcription of the beta subunit, which is 112 amino acids long. Again, this mRNA is translated in the cytoplasm, and together these two subunits are put together, essentially, in the Golgi apparatus. The Golgi then packages them up into vesicles, and these vesicles are stored at the surface of the cell. Now remember that IP3 increases calcium concentration in the cytoplasm. Here we see that calcium can increase the exocytosis of TSH, thyroid stimulating hormone, which again contains two different subunits. This is a protein hormone that is then released into the blood and ultimately is going to be taken out of the anterior pituitary gland and on towards the thyroid. Now, all of this is stimulatory. Again, TRH from the hypothalamus triggers the release of TSH into the blood in the anterior pituitary gland. The negative feedback of this system is induced by thyroid hormones. Okay, so ultimately, when there is an abundance of TSH is going to stimulate the thyroid to release T4 and T3. Okay? When there is enough, essentially, thyroid hormones, T4 or T3, these hormones are also going to be delivered in the blood to these thyrotropes. And T4 in particular can reduce the sensitivity of the GPCR to TRH. So TRH, even if it is still in the blood, it is not going to activate this entire system quite as much if there is already abundant thyroid hormone in the blood. Also, as we know, thyroid hormones can slip right through the membrane. Okay? Deiodinases within the thyrotropes or thyrotrophs can convert or activate T4 into T3, and this T3 it works to exert a negative inhibition, essentially, on the release of TSH by interfering with this transcription factor. So again, negative feedback is exerted by these thyroid hormones by directly interfering with the transcription factors and reducing the sensitivity of the thyrotropes to TRH. Once again, this has been essentially part 2 of the H hypothalamus, P anterior pituitary gland, and thyroid axis. Thank you.